Students, we are discussing other applications of biotechnology, right? Other applications of biotechnology. Yesterday, we discussed, right, introduction concerned to other applications of biotechnology, right? In this other applications of biotechnology, we are going to discuss about number one, insulin. What is the first one? Insulin. Number two, second one is gene therapy. What is the second one? Gene therapy. And third one is molecular diagnosis. What is the third one? Molecular diagnosis. Molecular diagnosis. Is it okay? Right? Go for the first one. What's insulin? Go for insulin. Right? Now, what is the meaning of diabetes? Diabetes means nothing but if the level of sugar in blood is more, then we are calling it as a diabetes. Yes or no? Right. Therefore, what is the meaning of diabetes? What is the meaning of diabetes? Right? Diabetes means nothing but level of what? Sugar. Level of sugar is more in what, sir? Blood. Therefore, if level of sugar in blood is more, we are calling it as what, sir? Diabetes. We are calling it as what? Diabetes. Is this statement okay? Right? Sir, this diabetes can be cured by using what, sir? Insulin. Because this insulin is a hormone which maintains the level of sugar in the blood. Is it okay? Right? Therefore, insulin. What, sir? Insulin, what it does? Maintains or controls. Maintains or what, sir? Controls what? Level of sugar. Level of sugar in blood. Who is controlling the level of sugar in the blood, sir? Insulin. Is this statement okay? Therefore, this insulin, what? Insulin used to control diabetes. Used to control what, sir? Diabetes. Is this statements okay? Right? Sir, from where this insulin is produced from? From where this insulin is produced from? Now listen this statement. Insulin production we are discussing, right? What production, sir? Insulin production. Insulin production. Sir, in early days. In what, sir? Early days. In early days, this insulin is produced from pancreas. Produced from where, sir? Pancreas. In early days, insulin produced from pancreas. Produced from pancreas of slaughtered pigs and cattle. Is it okay? Therefore, in earlier days, insulin is produced from pancreas of what, sir? Slaughtered pigs and cattle. But at present, nowadays, this insulin is produced right inside E. coli bacteria by genetic engineering process. Therefore, nowadays insulin produced where, sir? Insulin produced inside the bacteria. Insulin produced inside the bacteria by genetic engineering process. By which process, sir? Genetic engineering process. By genetic engineering process. Therefore, by genetic engineering process, nowadays this insulin is produced inside what, sir? Bacteria. Is this statement okay? Now listen this statement. Sir, the insulin which is produced inside the bacteria by genetic engineering process, that insulin structure is identical to insulin that is naturally produced by natural process. By the natural process, how the insulin is like that we are getting insulin right inside the bacteria. By which process? Genetic engineering process. Is this statement okay? Right? Therefore, insulin produced, what, sir? Insulin produced right where? Inside bacteria by genetic engineering process. By which process? Genetic engineering process is having what? Identical structure to what? Identical structure to insulin produced in nature. Insulin produced in nature. Is this statement okay? Clear? Right? Therefore, its structure is identical. Its structure is identical to the structure of insulin that is produced in nature. Is this statement clear? Right? Now listen here. Sir, insulin. What it is? Insulin. Sir, insulin is a hormone, right? Insulin, it is what, sir? Insulin is a hormone. Chemically it is a protein, yes or no? Chemically it is what, sir? Protein. Chemically it is a protein. Now listen this statement. Sir, this insulin, it is a hormone, sir. Chemically it is what, sir? Chemically it is a protein. Chemically it is a protein. What, sir? Insulin. Now listen here. Sir, this insulin, what it is? Insulin. Chemically it is a protein. Protein means made up of what, sir? Amino acids. Proteins means made up of what? Amino acids. Yes or no? Now listen this statement. Sir, insulin is produced as a prohormone. Insulin produced as a what, sir? Prohormone. There is nothing but it is produced as a proinsulin. It is produced as what, sir? Proinsulin. It is produced as what? Proinsulin. Now listen here. Sir, insulin is produced as a prohormone, right? There is nothing but what, sir? Proinsulin. Which insulin, sir? Proinsulin. Proinsulin, right? What it is? Proinsulin. Sir, this proinsulin, it contains what? It contains a polypeptide chain. A polypeptide chain, B polypeptide chain, B polypeptide chain, B polypeptide chain, B polypeptide chain, as well as a C polypeptide chain. Therefore, A peptide chain is present, B peptide chain is present, C peptide chain is present. In case of what, sir? Proinsulin. Is it okay? Right? Sir, A polypeptide chain, B polypeptide chain, C polypeptide chain, these are present in what, sir? Proinsulin. Present in proinsulin. During maturation. During what, sir? Maturation. Now listen here. During maturation of proinsulin to insulin, during maturation of what? Proinsulin to insulin, C chain is removed. Therefore, C peptide chain. What, sir? C peptide chain. C peptide chain is removed. What is removed? C peptide chain is removed when during maturation of what? Proinsulin to what? Insulin. Therefore, during maturation of proinsulin to insulin, what is removed, sir? C polypeptide chain, that is a C peptide chain is removed. It means now this fully matured functional insulin contains A peptide chain and B peptide chain, but not C peptide chain. Yes or no? Therefore, sir, this is what? This is insulin. This insulin is fully matured. Fully matured and functional. Sir, this fully matured functional insulin, this fully matured functional insulin contains what and what, sir? It contains A peptide chain and B peptide chain. What and what it contains? A peptide chain as well as B peptide chain. A peptide chain and B peptide chain. Is it okay? Right? Sir, this A peptide chain contains 21 amino acids. A peptide chain contains how many amino acids? 21 amino acids. Whereas B peptide chain, it contains 30 amino acids. Is this statement okay? A peptide chain is with 21 amino acids, whereas B peptide chain is with 30 amino acids. Now listen this statement. Sir, for example, this is a amino acid, sir. What is this one? Amino acid. Sir, this is another amino acid, sir. This is another amino acid. Therefore, in this diagram, how many amino acids are there? 1, 2, 3. 3 amino acids are present. In first year, we read that in between two amino acids, which bond is present? Peptide bond is present. In between what? Two amino acids, which bond is present? Peptide bond is present. See this diagram? In diagram, 3 amino acids are present, whereas number of peptide bonds are how many? 2. Therefore, what is the formula to calculate the number of peptide bonds? Number of peptide bonds is equals to number of amino acids minus 1. Yes or no? Sir, in this diagram, 3 amino acids are there. 3 minus 1, how many? 2. What are 2? Peptide bonds are 2. Therefore, to calculate the number of peptide bonds, the formula is number of amino acids minus 1. Number of amino acids minus 1. We are saying that A peptide chain is with 21 amino acids. Therefore, number of peptide bonds becomes how many? 20 peptide bonds. How many peptide bonds, sir? 20 peptide bonds. 20 peptide bonds. Sir, B peptide chain contains 30 amino acids. 30 amino acids means how many peptide bonds, sir? 29 peptide bonds. 29 what, sir? Peptide bonds. 29 peptide bonds. Is this statements okay? Clear? Right? Therefore, sir, fully matured functional insulin contains A peptide chain and B peptide chain. A peptide chain with 21 amino acids and B peptide chain with how many amino acids? 30 amino acids. Is this statements okay? Right? Now we are discussing what insulin. Nowadays this insulin is produced inside bacteria, we are saying. Produced inside bacteria by genetic engineering process. Yes or no? Right? Now listen this statement. In 1983, a American company by name Lilly. What is this American company name, sir? Eli Lilly. It is a American company. In 1983, Eli Lilly, a American company, right, produced or synthesized, right, A chain, produced what, sir? Produced 2 DNA sequences. Produced 2 DNA sequences. Produced 2 DNA sequences which codes for A chain and B chain, which codes for A chain and B chain of insulin. A chain and B chain of insulin. Sir, what is the meaning for this statement? First, listen this statement. Sir, in 1983, a American company by name Eli Lilly has synthesized or produced two DNA sequences, sir. In these two DNA sequences, one DNA sequence is coding for A chain of insulin, another DNA sequence is coding for B chain of insulin. Is this statement okay? Right? Now, listen this statement. Sir, insulin means what plus what, sir? Insulin means A chain plus B chain, yes or no? It means A peptide chain plus B peptide chain. A peptide chain plus B peptide chain together forms what? Together forms insulin. Together forms what, sir? Insulin. Sir, A chain, it is a peptide chain. Peptide means nothing but protein. It means a protein is synthesized from where, sir? It is synthesized from mRNA. Sir, B peptide chain. B peptide. Peptide means protein. Protein means synthesized from where, sir? mRNA. By which process? Translation process. Yes or no? By which process, sir? Translation process. Now you tell me, sir, how this mRNA is synthesized? mRNA is synthesized by transcription process. Synthesized by transcription process. Who undergoes transcription? DNA undergoes transcription. Sir, mRNA is synthesized by transcription process. Who undergoes transcription here, sir? DNA undergoes transcription. Yes or no? Right. Therefore, by which process, sir? Transcription process. By which process, sir? Transcription process. DNA synthesizes mRNA. DNA synthesizes mRNA by transcription. Now, by translation process. By which process, sir? Translation process. mRNA synthesizes a protein, right? Therefore, mRNA produced A chain by translation. mRNA produced B chain by what, sir? Translation process. Yes or no? Right? Now, see this statement carefully. In 1983, a American company by name Eli Lilly synthesized what, sir? Two DNA sequences. It means this Eli Lilly, it has synthesized artificially two DNA sequences. Sir, what are the two DNA sequences, sir? One DNA sequence codes for A peptide chain of insulin. Another DNA sequence codes for B peptide chain of insulin. Is this statement okay? Is this clear? Right. Therefore, in 1983, a American company by name Eli Lilly artificially synthesized two DNA sequences. Out of two DNA sequences, one codes for what? A chain of A peptide chain of insulin, and another DNA sequence codes for B peptide chain of what, sir? Insulin. Is this statements okay? Right. Then what this Eli Lilly American company did? Now listen. How many DNA sequences artificially they synthesized? Two DNA sequences, right? DNA sequence which codes for A chain. Is it okay? What is the second one? DNA sequence which codes for which chain? B chain. Right. What they did means they introduced this DNA sequence which codes for A chain into plasmid. Into what? Plasmid of what, sir? E. coli. Plasmid of E. coli bacteria. Plasmid of what, sir? E. coli bacteria. Is this statement okay? Clear? Right. Therefore, this DNA sequence which codes for A chain was introduced into the plasmid of E. coli bacteria. Plasmid of E. coli bacteria. Then what is formed, sir? rDNA is formed. rDNA. What is formed, sir? rDNA is formed. Sir, this rDNA is introduced. This rDNA is introduced into E. coli bacteria. rDNA introduced into which bacteria, sir? E. coli bacteria. rDNA introduced into E. coli bacteria. Sir, now here rDNA means what, sir? Vector plus desired gene. What is the vector here, sir? Plasmid. What is the desired gene? DNA sequence which codes for A chain. Sir, when this desired gene is expressed, it means when this DNA sequence which codes for A chain is expressed. When what? DNA sequence which codes for A chain. DNA sequence which codes for A chain is expressed. What is produced? A chain is produced. What is produced, sir? A chain is produced. Similarly, DNA sequence which codes for B chain. It was introduced into the what, sir? Plasmid. It was introduced into the plasmid of which bacteria? E. coli bacteria. Then what is formed, sir? rDNA is formed. rDNA is formed. This rDNA is introduced into which bacteria, sir? This rDNA is introduced into E. coli bacteria. Right, rDNA introduced. rDNA introduced where? Into E. coli bacteria. Is it okay? Now you tell me, where is this rDNA? Inside E. coli bacteria. rDNA means what, sir? Vector plus desired gene. What is the vector? Plasmid. What is the desired gene? DNA sequence which codes for B chain. It means now the DNA sequence which codes for B chain is in E. coli bacteria. If this DNA sequence which codes for B chain is expressed in E. coli bacteria, then what it produces? Produces B chain. Produces what, sir? B chain. Therefore, which chain is produced? B chain is produced. Therefore, A chain and B chain are produced separately. A chain and B chain are produced separately in E. coli bacteria. A and what, sir? B chains. A and B chains produced. Produced separately. Where, sir? In E. coli bacteria. Then now you tell me where A chain is produced? In E. coli bacteria. Now this A chain is isolated from E. coli bacteria. This B chain is isolated from E. coli bacteria. After isolating A chain and B chain from what, sir? E. coli bacteria. After isolating A chain and B chain from E. coli bacteria, in between this A chain and B chain, in between what? A chain and B chain, right? Disulfide bonds are established. What are established? Disulfide bonds are established. Is this statement okay? Right? Therefore, A chain and B chain isolated. A chain and B chain isolated from where, sir? E. coli bacteria. After isolating them from where, sir? E. coli bacteria, in between A chain and B chain, in between what and what? In between A chain, that is A peptide chain, and B peptide chain, in between A peptide chain and B peptide chain, what are established? Disulfide bonds are established. What are established, sir? Disulfide bonds are established. How many disulfide bonds are established, sir? See the diagram. 2 disulfide bonds are established. Is this statement okay? Clear? Right? Therefore, how many disulfide bonds are established, sir? 2 disulfide bonds are established. Disulfide bond. This is another disulfide bond. Is this statement okay? Right? Therefore, now in between A chain and B chain, what established? Disulfide bonds are established. Total how many disulfide bonds are there, sir? 2 disulfide bonds are present. Is it okay? Now this is called as insulin. Sir, what is here to call it as insulin, sir? A chain, B chain, linked by what? Two disulfide bonds. Called as what, sir? Called as a insulin. Called as what, sir? Insulin. Is this statement clear? Is it okay? Now listen this statement. Sir, this is which chain, sir? A chain. Within A chain, one disulfide bond is present. Within A chain, one disulfide bond is present. In between A chain and B chain, two disulfide bonds are present. Is this statement okay? Within A chain, one disulfide bond is present. In between A chain and B chain, two disulfide bonds are present. Sir, this is the insulin produced in E. coli bacteria. This insulin produced in E. coli bacteria is called as humulin. What it is called as a humulin? It is called as humulin. Insulin produced what, sir? Inside E. coli bacteria by genetic engineering process. Called as, what is that insulin called as? A humulin. What it is called as? A humulin. Now listen here. Sir, I am taking insulin, right? Sir, in this insulin, within A chain, right, one disulfide bond is present. In between what? In between A chain and B chain, where in between A chain and B chain, two disulfide bonds are present. Is this statement okay? Right? Now listen here. Sir, this insulin which is produced in E. coli bacteria, due to the production of insulin inside E. coli bacteria, there is a continuous supply of insulin. Number two, due to this insulin production inside E. coli bacteria, right, the market price of insulin was stabilized. Therefore, what are these two statements? Right, insulin due to production of what? Insulin by genetic engineering process, where inside E. coli bacteria, due to production of insulin by genetic engineering process inside E. coli bacteria, right, what happened? Continuous supply of insulin. Supply of insulin is continuous. Number two, what more? Market price of insulin. Market price of insulin stabilized. What is stabilized? The market price of insulin stabilized. There is a continuous supply of insulin, right? This is due to production of insulin by genetic engineering process. Is this statement okay? Right? With this we completed the first one. What it is? Insulin, right? Now go for number two, second one. What it is? Gene therapy. This gene therapy, right, it will come under zoology. Right, in detail you will get a discussion of gene therapy in zoology, right? Just we are introducing this topic in botany, okay? Right, what it is? Gene therapy. Sir, gene therapy is a corrective therapy. Sir, why we are calling it as corrective therapy, sir? For example, take a individual. Take a individual, or you take a embryo of an individual, right? Sir, in this individual many cells are present, yes or no? In this embryo also many cells are present, yes or no, right? From the cells or tissues of an individual, or from cells or tissue of what's our embryo, right, defective gene is removed. What is removed, sir? Defective gene is removed. And that defective gene, after removing the defective gene, we are placing a functional gene. We are placing a functional gene. Is this statement okay? Right, therefore what is removed, sir? Defective gene removed from cells or tissues of an individual or embryo, right? Sir, after removing the defective gene, right, from the cells, right, or tissues of an individual or an embryo, what we are doing? We are placing the normal gene. Placing the normal gene. It means non-functional gene is replaced by a functional gene. Defective gene is replaced by a functional gene. It means the mistake that was done is corrected. So this is called as what, sir? Corrective therapy. Is this statement okay? Right? Therefore, non-functional gene. What, sir? Non-functional gene or defective gene. Non-functional gene or defective gene is replaced by what, sir? Is replaced by functional gene. What is replaced by what, sir? Non-functional gene or defective gene is replaced by functional gene. So we are calling this as what, sir? Corrective therapy or what, sir? Gene therapy. Is it okay? Right.